All right, so far we have talked through an overview of photosynthesis as well as discussed how different reactions are able to harness the light energy. So now we're going to get into the specifics describing what's actually happening to the electron as it moves from photosystem 2 to photosystem 1, such that we can ultimately produce ATP and NADPH. All right, so again, just to orient you where we're at in the plant cell, we are inside of an actual chloroplast, and more specifically, we are inside the thylakoids. And what you're seeing here is the phospholipid bilayer of, let's say, the top layer of this stack of thylakoids. So here's one thylakoid, and now you can see the next thylakoid in that series. And so we're talking about what's happening in the phospholipid bilayer of the thylakoid itself. And so this space outside of the thylakoid we would call the stroma. That would be this brown part here. And inside of the thylakoid itself we call the thylakoid lumen. All right, so again, we already talked about what's actually happening to chlorophyll when it gets excited by the light. We talked about that vibrational energy, that transfer of resonance energy. And that energy is going to get transferred to an electron. So where's the electron actually coming from? Well, it's coming from a water molecule. So here's a water molecule, and it's going to be lysed. So we call this process photolysis. It's literally energy that's ultimately going to come from the light. So we call this photolysis. Is going to split that water, and we're going to have these two electrons that get released from breaking up this water molecule. Now, the electrons from the water molecule are in a low energy state, and those electrons are going to reside with a protein, or a chlorophyll, I should say, called P680. And P680 is named because it's a pigment molecule that gets excited by a wavelength of light at 680 nanometers. So upon activation, that electron that's residing with P680 ultimately is going to get activated, and we're going to see a major increase in the energy of those particular electrons. And once it's fully activated, P680 is then going to transfer the electron down an electron transport chain, just like what we saw in mitochondria with regards to cellular respiration. As it gets transferred from one complex to another, we're going to see a decrease in energy of that electron. And so the red marking here is showing the flow of that electron. Ultimately, once that electron ends up at this cytochrome complex, it's going to do some work. And the work it's going to do is going to pump some protons from the stroma across the phospholipid bilayer of the thylakoid membrane and dump those protons into the thylakoid lumen. So we're going to see a net increase in proton concentration within the thylakoid lumen. So again, anytime we're going against a concentration gradient, that's going to require some energy. Where did the energy come from in this particular situation? It came from the electrons. Where did the electrons get their energy from? They got it from the light. And where was that initial electron from? It came from the water. All right, so at this point, the electron has done some work, and so its energy is somewhat depleted. And so it's going to continue to get passed down this electron transport chain. And once it hits photosystem 1, we're going to see an excitation of the energy of that electron because there's a different type of chlorophyll here called P700, and P700 absorbs light energy at a wavelength of 700 nanometers. We're going to have a net increase in the energy of the electron here, and then that's ultimately going to get transferred to something called ferredoxin. Ferredoxin is then going to transfer it to the enzyme called NADP plus reductase. So anytime you hear oxidation and reduction, you should immediately think of who's losing and who's gaining the electrons. So reductase implies reduction is about to take place. Reduction is a gain in electrons. So where is this electron that originally came from water? Where is it going to reside? Well, NADP plus is going to pick up that electron and form something called NADPH. And so you can think of this as a high energy electron carrier. The electron that it's carrying has a lot of energy. Where did that energy come from? It came from P700. So at this point, we now have a high energy electron carrier, which we will utilize in the Calvin cycle. But in the meantime, we've done something which is increase the proton concentration in the thylakoid lumen. So how did we increase the proton concentration? First of all, when we lysed the water, we saw a net increase in the amount of protons inside the thylakoid lumen. Secondarily, when the electron was getting transferred down the electron transport chain at the cytochrome complex, it did some work and it transferred those protons against its concentration gradient. So that's a second way that we increase the H+ concentration. And lastly, when we formed NADPH, we went from NADP+ with 2 protons to NADPH with 1 proton. So that decreased the H+ concentration out here in the stroma. So those 3 things together are all going to cause you to have a high difference in concentration in the thylakoid lumen versus in the stroma. So left to itself, those protons ideally want to equalize the proton concentration on either side of this membrane. However, they are not permitted to cross the membrane because they are charged molecules. So the way that they are going to move out of the thylakoid lumen is via ATP synthase. And so again, this should be very reminiscent of what we talked about with regards to cellular respiration. It's going to work just the same way as what we had already talked about. Those protons are going to flood through ATP synthase. Through that proton motive force, we're going to see ATP synthase start to move, and that is going to result in the formation of ATP. Okay, so I'd quickly like to describe to you what is actually happening to the energetics of that particular electron, again, that originally came from water itself. So water is going to be split by a process that we called photolysis, and that is going to hang out and reside with P680 in this unactivated state in what we call the reaction center. Eventually, a photon of light is going to excite one of those pigment molecules, and via resonance, transfer of energy, that vibrational energy, one chlorophyll or one pigment molecule is going to activate another until ultimately that energy is going to result in the vibrational energy and activation of P680. The electron, which came from water, is now going to get activated and able to absorb that energy and is going to move into this highly activated state. It's then going to get, the electron is going to get transferred down the electron transport chain. Once it reaches that cytochrome complex, that's where we're going to see the pumping of protons against the concentration gradient. And then we're going to see a decrease in energy because it just did a bunch of work until it hits that photosystem 1. And again, light energy is going to result in the activation of that electron getting re-excited. That will then get transferred to ferredoxin. Ferredoxin is going to transfer the electron to the enzyme NADP+ reductase. And NADP+ reductase is going to cause the transfer of the electron from NAD to NADP+, forming NADPH. So you can see that we had a low energy electron, it got activated, it did a bunch of work, we had to reactivate the electron until it ultimately resides with NADPH, which we can call a high energy electron carrier because the electron that it is carrying has a lot of energy. Where did the energy come from? It came from light. Where did the electron come from? It came from water. So what is actually produced by these light reactions? Well, first of all, it's important to point out that oxygen is produced. When that water gets lysed in the photolysis step, in the thylakoid lumen, we are actually producing oxygen. That's taking place at that photosystem 2. And again, in so doing, we then transfer 2 electrons to the P680 molecule that's residing in the photoreactive center of photosystem 2. We also produce NADPH. This is going to be produced not in the thylakoid lumen, but rather in the stroma. And this is where those high energy electrons are ultimately going to reside. They get activated ultimately at photosystem 1 by the P700 and then get transferred to. Lastly, we're going to form ATP. This is due to ATP synthase, which is residing in the thylakoid, the phospholipid bilayer of the thylakoids themselves. And ATP is going to be produced in the stroma. And this is a result of that proton gradient that was generated via 3 main mechanisms. Okay, so it's also important for you to remember where are each of these products produced. So again, the oxygen is going to be produced inside the thylakoid lumen. The NADPH is produced in the stroma, and the ATP is also produced in the stroma. So if you were asked how is ATP synthesized in chloroplasts, previously we've already talked about substrate level phosphorylation and we have also talked about oxidative phosphorylation, but this really is neither of those. We call this process photophosphorylation, and that is because if you really look at the water, what's happening to the water? Is it oxidized or is it reduced? The water itself is actually oxidized. It's losing an electron. And so because of the loss of that electron and ultimately the result of the excitation of that electron due to light, we call this process photophosphorylation. It's really due to the chemiosmosis that we ultimately are forming ATP. And so you can now add this third mechanism of how to produce ATP. In addition to oxidative phosphorylation and substrate level phosphorylation, you now can describe photophosphorylation as well. Lastly, one more thing you should be able to describe on your own is how is it that we are creating that proton gradient so that we can make the ATP. There's 3 different mechanisms. The first is an increase in the H+ concentration when that water is getting split. This is the photolysis. So here you see an increase in the H+ concentration. Secondarily, as the electron moves down that electron transport chain, we see an increase in the H+ concentration as that electron is doing work. We see a high concentration here in the lumen. And lastly, as we cause a decrease in the H+ concentration out in the stroma when we are forming NADPH, that also leads to a difference in the proton concentration on one side of the membrane versus the other. All of that is going to result in a huge push for those protons to move from inside the thylakoid lumen out into the stroma. That is going to cause that proton motive force, and the ATP synthase is going to end up resulting in the formation of large amounts of ATP.